and welcome to our continuing MICD webinar series for this year. It's my great pleasure today to introduce Denise Kirschner, a friend and colleague. Denise is a mathematician. She obtained her PhD from Tulane University and essentially has been at Michigan ever since. The interesting thing, of course, about Denise is that she's a mathematician fully embedded in a medical school department. Denise has always been at Michigan. She's been at the Department of Microbiology and Immunology. What Denise does is develop really complex multi-scale models of host pathogen interactions with application to a number of infectious diseases. And in particular, her work is concentrated on tuberculosis and HIV. Her aim is to understand how these complex dynamics and perturbations lead to the evolution of the disease over short to very long timescales. And importantly, her approach is truly multi-scale in time as well as in sort of a spatial scale, going from molecules to cells to systems. Denise is very well recognized in the field. She has long been editor-in-chief of the leading journal of theoretical biology. She was president of the Society of Mathematical Biology until 2020. And at Michigan, she is the founding director of the Center for Systems Biology, the founding co-director, but this is really is Denise's vision. Importantly, Denise has a great passion for fairness and equity in science, and this is something she brings to all her work. So we're very pleased to have Denise, and this has been long overdue. I've known Denise and been working with her for years and collaborated with her on various things. So it's really a pleasure, Denise, to welcome you to give us this talk in the MICD seminar series. Over to you now.
Thank you, Krishna. Thank you so much. It really is a pleasure to be a part of this today. And thank you so much for the invitation. And I just have to say, Krishna, we started out together really in the development of that Center for Systems Biology, and to see what you've built with the MICDE program is truly astounding, and how successful it has been. And so I greatly admire what you've done, and the outreach actually that it has had, the impact is really tremendous. My students and trainees have benefited tremendously from all the things that you've offered. So I'm grateful for that program, and we've even partnered together with the two centers and done some great seminar things. So it's been a long and wonderful history together. So I'm excited to be here today. So I'm going to go ahead and share my screen now, and let's see, how do I share? Here we go. And I'm going to share that, and just sort of start with some ideas from Descartes, which is my daughter's doing the Enlightenment in school now, and I came across this, and I thought it was pretty appropriate that, yes, because of COVID, we're doing this over Zoom, but there are some benefits we were talking about earlier, and that is that we can record these. We can also do it from all over the world. So it's a real pleasure to be here. So what I'm going to talk to you about today are approaches that I use for studying multi-scale computational models. And this is really important because actually these models are relatively new on the scene, and trying to come up with ways to analyze them, study them, and get useful information from them that can then be translatable into the experimental side of things is extremely important. And so here I'm showing you just from our system, we're interested in studying TB. Here's some lungs from a monkey. Here's some tissue from a monkey that's being studied immunologically. Here's our computer simulation, and all these things come together. So I think I'm going to start by telling you what I think multi-scale modeling is. And multi-scale modeling refers to a style of modeling where you have multiple models at different scales that are used simultaneously to describe a complete system, very complex biological system typically. The different models usually focus on different scales of revolution, and I'll explain that a little bit more. The reason that we need multi-scale modeling is because these problems are incredibly complex, and you really can't capture everything at a single scale. So for example, if you're interested in studying something within the human body, you're interested in maybe how the molecules work, but of course they're dependent on the genes and the cells that those molecules are within. And then those cells, of course, are made up of tissues which fill out organs. So you have many scales, but of course these scales, and many of us as modelers live at a particular scale. And what I want to argue to you is that the multi-scale framework is extremely important to be able to thread them and then be able to ask questions where potentially you vary something at the gene scale and it reads out on an organ or a whole host scale even. So that's our goal today with the talks that we're doing. So the issue is when you want to build models, there are different time and length scales associated with each one, and there's usually a different modeling type that might be appropriate at that particular scale. So if you start at the genetic or the molecular scale, the time scales, of course, are very fast, and the length scales are very, very short. And sometimes it makes sense to use sort of more deterministic and continuous dynamics. And believe it or not, usually there's statistical models that are capturing many of the things happening at the genetic scale and ordinary differential equations for things at the molecular scale. Intracellular and cellular dynamics are happening a little bit faster time scales, a little bit larger length scales, again, sort of behaving in a deterministic continuous way. And we can use things like ODEs to do that. At a tissue scale, again, we're even larger time scales and even larger length scales. You might want to capture things happening in a discrete stochastic fashion there. And so we use things like agent-based modeling, also known as individual-based modeling, where you're looking at individual agents that are moving around in space. And then, of course, the organ or the whole organism scale, which, of course, is a much longer time scale. If someone lives for about 80 years on average, and so you might be interested in that whole time length as well as the whole size of the person, and of course you could look at that deterministically, or you can look at it also discretely. If you're looking at a whole population, you might want to not compartmentalize, but you might want to track each individual, and so you would use a hybrid approach potentially there, depending on what the questions are that you'd like to ask. So these things are really important to consider as you build models that are multi-scale, is that each scale may have its own flavor type model. So if you decide to thread them together, you're going to have to be able to link models that are of very different flavors. So our approach typically is to start building the model at what we call the mesoscale, which is the middle scale. And then you constantly iterate and build that model with data. And then you have to choose an appropriate method, of course, and we've done some work on that. I'll explain it to you. And you calibrate and validate your model at that level and explore any emergent behavior there. So that's extremely important, in that each model you build, you want to really understand it at that scale before you build to the next scale. Then you need to link these models in space and time. And I'll tell you that there is no sort of short way method to do that. Many times it's linked through the parameter passing, but that's not always the case. There's other ways that you can pass this information. And you usually assume that the information flows in one direction, maybe from the molecular scale is feeding up to the cellular scale, but of course you need feedback as well. And so how those molecule concentrations, for example, are at one scale might depend on how the dynamics are happening at the scale below that. And so you need both the feed forward and the feedback information. And then we perform sensitivity and uncertainty analysis on these models, which is our traditional link to dynamical systems theory, which I'll talk to you a little bit about. And then we create models with tunable resolution characteristics, which I will also explain to you. Okay, so what I'm going to do is just have this tuberculosis model that we use as a touchstone for as I walk through these different multi-scale modeling approaches and methods that we do, so you can sort of see how it applies as we go. So we're very interested in tuberculosis, and we've been studying since 2001. And so the idea here is that TB is a bacteria that infects the world. And right now, 1/3 of the world is infected with TB. So that's a huge number, right? We're talking 2 billion people, right? And so the problem with that is that there are a large number of people who actually have active disease and die from it. And that's somewhere between 1.5 and 2 million deaths per year over the past decade. Now, that's more than COVID. And it is another global pandemic that exists in our world, although we don't see a lot of it in the Western world. The burden is typically in the Eastern world, and that may be why we're sort of immune or blind to it. So let me explain. When you get exposed, you breathe in bacteria, and then some percent of people breathe it in and clear it and are never infectious. A very small percent breathe it in and have active TB, where if they are not treated, they will die. And 90% of people are able to control it but not clear it. And the reason why that's important is because over your lifetime, that may reactivate to the primary active TB. okay, which is a real problem because that's the infectious one. And if you have 2 billion people that are in this latent state, any one of them could reactivate to active disease. And so different things can lead to reactivation, such as aging, which kind of wanes your immune system over time. Also, things like HIV, immunosuppressive drugs, malnutrition, all of these things can sort of weaken your immune system and allow for reactivation of this disease. And so, but of course, some people may never reactivate. So what happens is, oh, let me go back for just 1 second. This is a micrograph of a monkey, which this is a PET CT scan. So these are the same kind of scans that humans get. We have a PET CT in the facility where we can x-ray these monkeys, and you can see that these are granulomas that are forming within the lungs of these monkeys. So you get little foci where the bacteria land, develop something known as a granuloma. So I'm going to talk more about that soon. And this is how they develop. So you breathe in the bacteria, and then after a while, your local macrophages and dendritic cells take it up. But those macrophages are not able to clear the bacteria. Usually that's what they do. They chew it up, spit it out. Macrophage means big eater, and that's their job. But because we've been breathing in mycobacteria for thousands of years, they've built up an immunity to being killed that way. So eventually dendritic cells go to the local lymph node of the lung and stimulate T cells to come back. And when those T cells come back through the blood into the lungs, they are actually now able to activate those macrophages that were here, which can then clear the intracellular bacterial load. And they can also kill infected cells and through many different mechanisms, direct killing or through secretion of molecules that will do the killing for you. But the interesting thing is it kind of leads to the formation of this granuloma. And this is because mycobacteria grow very slowly. The doubling time of bacteria like E. coli is about 20 minutes, but for Mycobacterium, it's 24 to 48 hours. So that slow growth is actually like a virulence factor. It cloaks the bug. So much has developed by the time the T cell immunity shows up that it's never really able to completely get rid of it. It can just wall it off. And of course, you breathe in multiple bacteria, so you get the development of multiple granuloma. I'm also showing you here that there are factors like what I'm highlighting here. Tumor necrosis factor is one molecule, but there are many other molecules that are involved in this. I'm focusing on that because I'm going to show you an example of something later. So there's molecules, there's cells, there's bacteria, and there's multiple organs involved. Clearly, this is a highly complex problem that needs a multi-scale approach. This is just to show you that the granulomas come in many different types. And so here's something called a caseous necrotic. Much of the center is dead tissue. Here it's full of different types of dead cells. This was dead macrophages. These are dead neutrophils. This one is calcified. And this one doesn't have much caseum. It just has a lot of cellular debris. So the monkey model that we use, we have a colony of 300 monkeys housed in a biosafety level 3 facility at the University of Pittsburgh, where they have this work, my collaborator Joanne Flynn, which I'll talk some about in a little bit. And we use this model of cynomolgus macaque, where we infect it with a very low dose. So again, if you get food poisoning, you take in some bacteria, it's usually about 10 to the 6th, 10 to the 8th even for your inoculum. Here, it may even only take one bacteria to lead to TB. And there's many different ways we study these monkeys. One thing that's very interesting is that I told you that 1090 number before for humans. In the monkey model, it's about a 50-50 split between active and latent disease. So that allows us a larger N size to be able to study these two different types. So this is how we view it as this multi-scale model. There's molecules such as this tumor necrosis factor I showed you earlier. There's different dynamics happening there on the cellular scale. There's the different cells on the molecular scale. There's the different cells, the molecules, as well as the cells that are interacting. They read out at the tissue scale, which are all involved at the organ scale. And of course, each one of these is within a specific person. So our modeling approach has been, what is the most appropriate way we should look at this? You know, as I mentioned to you earlier, we build these separate models and then we thread them together. So where do we begin, and what's the appropriate model to use to develop that? You know, there's no right answer, I'll tell you, to begin with, but you just have to really think about the questions that you're asking when you approach a model like this. So We started out with an ODE model. Why? ODEs are relatively easy to study and analyze and simulate on a computer. And so we started there and we learned a lot. And then we did a PDE model because we realized that space was important in time. But PDEs are, as you know, are very hard to manage and study and solve. So we moved to something called a metapopulation model, where we build a fake grid, and inside of each one of those grids, we're running all the ODEs that were in our original model. That's kind of like a fake spatial model, and it's known as metapopulation modeling. It comes from ecology. And then we finally moved to agent-based modeling, where we started building agent-based models where we're using discrete and stochastic simulation to do this. And interestingly, we wrote a paper comparing these different methods and what you get from using these different kinds of approaches, and which one might be more appropriate depending on the kinds of things that you're looking for. So you can find that paper if you're interested, and all our papers are available on our website. Let me tell you that now today, whenever we're building an agent-based model, we actually always start with an ODE model, because the ODE helps us to write down all the equations of things that we're interested in, and also parameterize things a little bit more straightforward. Then we tend to translate those ODEs into agent-based modeling to help us with that transition. You don't have to do it that way. That's a way that we have seen that works quite well for us. So what is an agent-based model? For any of you that haven't really spent some time using these, I'll give you a brief primer here. So there are 4 things that we believe are important for agent-based modeling. First is the environment. What's your modeling space that you're interested in? So for us, it's lung tissue, and granulomas grow to about a couple of millimeters in size. So we start with a 2 millimeter, now we're up to 4 millimeter size square of tissue that we start with. And then we discretize this into grid compartments that are 20 microns by 20 microns inside. Why 20 microns? Because that's the size of our largest cell type in the model, the macrophage. And then you define the agents. Oh, and I should say, in our environment, we also have vascular sources, because the lung are highly vascularized. You need a lot of blood flowing in there. And so we're able to add these vascular sources in. So the next thing is, what are the agents that are involved? So our agents are the cell types, the different immune cells, the macrophages that are involved, and different types of T cells of different subtypes, as well as things that are continuous. So for example, cytokines and chemokines are molecules that diffuse within our grid. So we track those over time. Those are things that actually tell cells what to do, how to move. And then we also have the bacteria, which we can track in either a continuous way or in a discrete way, tracking every individual bacteria. We have a choice there depending on the questions that we're asking. There's 2 more important pieces when you build an agent-based model. First are the rules and interactions of how these agents interact with each other and the grid. That's usually the hardest thing, and that's where we draw from the biology the strongest. And then the time steps of these processes. You can imagine diffusion is happening on a much faster time step, something like seconds, to something like cell movement, which is happening on a much a slower time scale compared to that. So all of those get put together, and we build our model. So here's an example of what the rules would look like. So here's a cell that's secreting a molecule, and it happens in a specific way in our model. I'll actually talk about diffusion later. If a macrophage hasn't been activated by a T cell and it fills up with bugs, it can burst open and spread those bacteria around it. All of these are probabilistic events based on things that are known from in vitro and in vivo studies, and also from the monkey data that we have. There's also, here's an activation of a T cell, and there's thousands of rules in our model. But here's the amazing thing. We put these rules in at the cellular scale, and then we run the simulation. And when we see a granuloma form, it is an emergent behavior. We have not anywhere in the rules told it to draw a granuloma. That is a natural occurrence of the way the rules interact. And it also tells us that it is an attractor, and it's a stable attractor, because no matter how we run these models and the parameters that we put in, we actually get this granuloma every time. And so here's an image of what an actual 2D slice of a granuloma from a monkey looks like. It's been stained for 3 different cell types. And then there's that dead tissue in the center that happens from those initial cells that come and burst open and are full of bugs and they die. That actually kills the surrounding lung tissue. And so you get this caseum center. And then you notice that the cells are actually in this ring that forms, which is really amazing, but it's from the way that cells get recruited to this granuloma. So you can see that there's a ring of neutrophils, there's rings of macrophages, and then a cuff of T cells around the outside. Incredibly characteristic of the way these granulomas look. And if we simulate it, we also get a very similar sort of behavior with dead tissue in the center, surrounded by macrophages that are activated in the different states, resting macrophages that have been recruited, and the little pink and purple cells are the lymphocytes that were recruited. Now, of course, they didn't need me to draw this picture. They already have this information by their image. So what can we contribute now that we have a model that recapitulates this behavior? So the first thing we can do is we can look at it over time. To get that picture before, they have to sacrifice the monkey, take the granulomas out, slice them open, and stain them and look at them. But we can watch this evolve in time and say what happens as this granuloma forms to get to that place. Additionally, we can provide them with a time-lapse simulation of what that looks like. If you look carefully, you can see the diffusing molecules in the background there, and you can see this granuloma evolving over time. Now, of course, as modelers, we're able to tweak those parameters in the model, which allow different outcomes. Remember, at the beginning, I showed you multiple types of granulomas that they see. We can do a similar thing. The important thing is, though, that we're able to validate on different data that we use to calibrate the model to test that our model is actually making the right predictions. So what you see here are monkeys that have been necropsied over a 300-day period and looking at the bacteria load known as CFU for colony forming units over time, and that we're matching that data quite well. And now we have even more data available than what's on this graph. So we're continuing to match and refine that extremely well. And you can see that we always provide a standard error on because we can see a range of outcomes. It's not just a single outcome that we're going for. So I want to talk to you about some recent work done by a graduate student in our group, Louis Jocelyn, who has created a method we call CaliPro. So what CaliPro does is allows you to figure out how to calibrate your data, calibrate your model to data. So the first thing you have to ask yourself is, what are you calibrating to? If it's just a single trend outline, you can use some sort of optimization and an objective function to do that. There's multiple tools out there for that. If you have a range of data that you're trying to match and many time points for that, you have to ask yourself another question. Do you feel comfortable assigning a distribution to the observed data? If the answer is yes, then you can use Bayesian calibration techniques like sample importance resampling and other things like that. If the answer is no, then you can use our method CaliPro, which I'll walk you through here very briefly. And we have a paper, if you're interested, that's been published. You can find it on our website or you can search for it in PubMed. So the idea is you have a model and some estimates, and you've built your model. And now what you want to do is put it through this message to get a calibrated model that fits your data, multiple data sets in the end. So we start with putting inputs in, which are ranges on your parameter values. Every one of your parameter values in your model, you can assign a range to. And then you sample your, and you run your model, do many evaluations, and you decide some pass criteria and some fail criteria. And so then you continue this process where you continue to iterate and choose which parameter ranges allow you the pass criteria until you settle on a range of parameters that gives you like 95 or above in your fitting to the data that you have. And you can do that for multiple data like T cells, macrophages, bacteria load simultaneously. Okay, so the next thing I want to tell you is you have to think a little bit about dimensions. So, of course, the world around us exists in 3D and not in 2D. And we tend as modelers to model things in 2 dimensions because, one, it's easier to do, it's easier to simulate, right? 3D simulations take up a lot of time and energy and effort and computational cost. And so, of course, as we've been publishing this, people ask us, well, granulomas are really 3D structures, so can you model them in 3D? And the answer is yes, we can model them in 3D. So here's a simulation showing our 3D granuloma formation. I've made all those green macrophages on the outside transparent, so you can look through them to the center of the granuloma as this forms. Otherwise, you would just see a solid ball of green here. And you can see that we can model this in 3D. Now, this simulation takes about 7 days, as opposed to the regular 2D Grand Sim, which runs in a couple of hours. Now, this is a huge problem because I'm going to talk to you soon about running uncertainty and sensitivity analysis, and you need to run thousands of simulations to do that. So if you have something that takes 7 days to run, you really better know when do you need to run your 3D model versus when can you get away with running 2D. One of the nice things about running this 3D model, though, is that we can actually look at the molecules using something known as level sets. So we can, here's our granuloma, and here is the center of the granuloma, and you see that the level sets are little circles around each of the cells that are producing very small concentrations or very large concentrations of an amount of, and I'm using TNF here again, that tumor necrosis factor molecule. And we've used this visualization using Data Tank, which is freeware. So if you like what you see, there's many things Data Tank does for 3D simulation. That's fun. Here I'm going to slide this level set through the granuloma. And if you watch it, it becomes red and it shows you, it shows you the concentration on that slice. There it starts to show up in higher concentrations. You can see the concentration gradients on that level set as it passes through that granuloma. So there's a lot of beautiful ways that we can use it to help us what's going on, but it doesn't provide mechanism necessarily. We need to do analysis to get at mechanism. Okay, so let's talk a little bit more about the 2D versus 3D issues. So as we said, most things in biology are 3D, but most people model in 2D. So how wrong is that? Or how okay is it? So we've asked that question 2 ways. The first way is, when are we safe to use a 2-dimensional model? And the second way is, what if we have a 2D model, but the data that our experimentalists give us or that we've generated is from 3D? How do we scale that data to 2D? So we've looked at that in 2 different ways, both the role of dimensionality and how to scale the data back. And that's pretty recent work from our group in the past, few years. So when can you coarse grain and simulate 2D rather than 3D? And it's based on a couple of things. The first is space limiting crowding effects in 2D versus 3D. So there's a very famous problem that's been around for 50 or more years where you have someone who is at a bar and they get drunk. Now, if they're in 2D, they can eventually find their way home. But if they're in 3D, they will never find their way home. And so that's actually true, that the complexity when you move from 2 dimensions to 3 is incredible because you have 8 choices in your Moore neighborhood to move on your grid in 2D, but you have 27 ways to move if you move to 3D. And so that alone changes the way cells move and interact with each other. So if you're thinking about mechanisms where cells or molecules have to find each other in space and time, then you really need to be in 3 dimensions. But if that's not the focus of your questions, it's probably okay to do a 2D model. Now, the sensitivity of model parameters that determine 2D versus 3D, we did that both to see which were coming up. The only ones that were really different had to do with cell movement and cell recruitment to the granuloma. And so that's really important because in immunology, cells have to actually locate to communicate. Sometimes they communicate through molecules that distribute, but sometimes they communicate by being side by side. So if those are the questions that are important, and again, I'm using cells as the example, but that could be whether you're talking about rabbits eating hares or whatever you're, no, actually it would be foxes eating rabbits. That would be an important thing to think about. So if you're trying to model that, and you would need a 3D setting to answer those questions extremely well. And so whether you use 3D or 2D really depends on the questions that you're asking. Okay, so now what I'm going to do is talk to you about how we use these models. Because if you think about it, again, the point of it is not to recapitulate just what they see. They already get that data from the animal models. So why do they need us? They need us because we can help them identify mechanism. And the way we do that is through the use of uncertainty and sensitivity analysis. So uncertainty lets you know how much variability comes out of your model depending on what you put in. And that's basically the parameters and initial conditions. We use something called Latin hypercube sampling for that, but I'll talk about that in a little bit. And then also sensitivity analysis as well. In those parameters that you put in that gave you variation out, who's contributing the most to that variation. And that allows you to identify which are the parameters that are your handles for turning your system sort of up and down. And we use something called partial rank correlation to do that. If you're, there's also other approaches too, which I'll mention. But the neat thing is that we can quantify this over time, because which parameters are important maybe early on in granuloma formation might be very different later in maintenance of these granulomas over the lifetime of a host. So that time piece is extremely important. And we published a paper on this over a decade ago, and we have all programs to do this available in both MATLAB and R on our website that you're welcome to use. And so here's how we do uncertainty. Excuse me, just a second. We define a range of parameters. Sorry. We defined a range on each of our parameters, K in this example. And then we look at a hypercube of our parameter space. And then we're going to slice up our parameter space and choose a number of samples. Now, as you increase your sample size, you get better accuracy. There's a rule of thumb on how to do that. We use about 1,000. And so that actually tells you how many times you have to simulate your model. And then you need to divide your parameter space into equal probable intervals. And so to do that, here's an example. So I've taken my parameter 1 and my parameter 2, and I've given it a range here. And I'm dividing it n times. And I don't know anything about this, so my probability density function is just going to be a uniform density function because I don't know. So my equal probable intervals look like this. So I'm going to sample. First parameter A will be the blue choices, then my second sample will be B, where I choose from parameter 1 and 2, then my third one will be C, and so on, and I create a matrix here. And now I'm going to simulate my model n times with each vector of this sampling space that I've done. Then for the rank, and then I'm going to simulate it. Now for the rank correlation, I'm going to run this simulation and define the number of outputs that I care about. So is it bacterial load? Is it how many macrophages or T cells are in the model? Whatever that output is that you're interested in. And remember, standard correlations quantify linear correlations between variables, but these models are a highly complex nonlinear, but they have to be monotonic to be able to use partial rank correlation. And so instead of ranking them by their linear correlation, we rank them according to a defined correlations 1, 2, and 3. That's the ranking. And then they have to be between -1 and 1, of course. And I want to tell you that these are traditional transcritical bifurcations that you would necessarily find from doing dynamical systems theory. You can sort of notice that that happens, although we haven't shown that sort of theoretically, but we believe that that's what it identifies. And you can do this for every time point in your model. And so the other thing is, I mentioned earlier another method called EFAST, because what if you have an oscillatory model? That certainly is not monotonic, but we have another method called EFAST, which allows you to do that, and that will identify things that are more like a hop bifurcation. So here's something to think about. In a deterministic model like ODEs, you put your input, which has some uncertainty, as we said, the parameters, you run your model, and you get an output, which creates uncertainty. But in a stochastic model, as you put inputs in, you also pick up other uncertainties by stochastic effects, like the probabilities that are part of your model. And so that contributes in a different way to the uncertainties. So when you do the simulations that in these kinds of models, you also have to worry about stochastic effects and do another kind of sampling. And so there's epistemic uncertainty is the first type. Aleatory is the second type. So we have to add another set of iterations to your model to be able to capture that. The other thing that's really powerful about this uncertainty and sensitivity analysis is that it allows you to do, let's say you have a blood compartment and a lung compartment. You can do the sensitivity analysis in one compartment, within another, or between compartments, or do it in this compartment and see what the readout looks like in that compartment, and vice versa. So it's a very powerful for doing inter and intra compartment analyses. And that becomes important when we have these multi-scale models. You can tweak something down here and say, how important is that affecting this part of the model up here, or vice versa. Okay, so when I ran our sensitivity analysis, this is what I got for the GrandSim model. And you notice, and I'm only showing you the parameters that showed up as significant. You notice that in the first couple of days here, as that granuloma is forming, so maybe the first 40 days, the parameters that are affecting things early on are somewhat different than the parameters that are maintaining this granuloma structure over time. And that's extremely important because it tells you that the things that take to create this granuloma are very different than what it takes to maintain it. So that's why looking at a sensitivity and uncertainty over time is extremely important. Also, I want to tell you now, this is really important to think about. Many people that do modeling vary one parameter at a time. Well, now I'm going to study in this model what alpha does. And so you hold everybody fixed and you vary alpha and you say, look what alpha does. But remember, in your parameter space, you fixed it in one region when you do that. So what if you were over here and you fixed alpha? And that's a local sensitivity analysis, what you've done. Doing a global sensitivity analysis, which is what this provides you, is extremely important because you have to vary all the parameters simultaneously. You can't just vary a single parameter because that just tells you a little piece of space. Sure, you varied alpha over time, but beta was set to 20. What if beta was 10 and you vary alpha? You might get a completely different output, which is why we suggest that you vary all your parameters simultaneously in a global way. Okay, so if you look at my output here, what you find is that really remember there's hundreds of parameters in my model. The ones that are significant are actually quite few, and they tend to bin in 5 ways. These types of different rates in the problem. I'm going to focus on TNF. TNF seems to be important early on here. It's showing up a couple times early, showing up here, and also it's showing up all throughout the process from beginning to end. So that helps me to be able to drill down. So I'm going to stop here and take a little sidestep into sampling schemes really quick, because this is some new work coming out of our group. So remember I showed you we use this Latin hypercube sampling to sample our parameter space. What if that's not the best method? What if just random is better? And there's also more deterministic sampling methods called Sobol sequences, which might be better. So here I'm showing you an example of a random Latin hypercube and a Sobol of just a set in MATLAB that we did really quickly, where we just covered a space. There's something really obvious you should notice right away, and that is that random, is missing lots of holes in your parameter space of interest. Latin hypercube does a little bit better. There's still maybe a little hole up here in the corner. But look at this deterministic method, Sobol. It covers it completely every time. And because it's deterministic, you can repeat it every time. All right. So what about the computational time of doing this kind of sampling? Well, you notice that LHS takes the most time, whether you're in 2D or 3D, and random and Sobol take much less computational time. So maybe there are other sampling schemes which would do better. So I just wanted you to think about that. Oh, sorry. Okay. So we can compare these different types of schemes. Random, of course, is easy to implement, but it could be inefficient. Latin hypercube, it covers more uniformly than random, but it's difficult to add samples later, and it's also a higher computational cost. Sobol is reproducible, which is fabulous. It's very efficient, but it may not be suitable for non-uniform distributions of your parameters. So we took a very simple ODE model. It's an SIR model. It's not necessarily important here, but it only had 3 parameters in it. And so we said, what if we look at the sensitivity, the uncertainty here. And so you can see that they all converge after 1,000 samples to pretty much the same significant value. But if you use lower numbers of samples, you can see that there's wide variation on random and LHS, but the Sobol dives right in. So Sobol sequences may be a quicker, more accurate way, especially if you have a few number of samples that you can run your model. Because, this is a simple ODE model, runs very fast, but as I told you, GrandSim takes hours. 3D GrandSim takes days to run. So we might choose a Sobol sequence there for running our LHS as opposed to LHS. For running our sampling as opposed to LHS. We also looked at the average error. there. And you can see that random has the most error over time, and Sobol has sort of the least amount of error over time, and LHS is somewhat in the middle. They're all still pretty relatively small though, which is comforting. So none of them are bad. It just really has to do with kind of preference and computation challenges. Okay, so let's see. Let me see what time it is. So I am going to now tell you, if we relook at the sensitivity analysis I showed you earlier, it may be that early on, these very small sensitivities that showed up might actually not have shown up as significant during Sobol. So we're reevaluating that now. So Sobol was a little bit more accurate and efficient, and there were things that showed up when we did this using uncertainty and sensitivity analysis. So it may be in the end that Sobol is the best way to go. So we're still right in the middle of this, getting ready to publish this paper. But of course, the reason I was doing this sensitivity is because I want to report to the experimentalists what is the most important handles in this model that we can drill down and study further. So as I told you, T and F dynamics are really important here. So let's look at the original GrandSim model. Here I've simulated this granuloma, and now I've simulated it where I knocked out T and F from the beginning. So suppose I remove T and F from the system and rerun my granuloma. You can see what happens. You have tons of bacterial load in the center, and over time, this granuloma actually even falls apart and breaks up and begins to spread into the airways. So that tells them that TNF is very important in your system. So why build a model that recapitulates what we already know? Because experiments actually already know that TNF is really important. They have mouse models that if they knock it out or they deplete it from the system, then the mouse gets active disease. And the same with the monkeys, and even in humans, if they take drugs for rheumatoid arthritis and things, it actually takes all the TNF out of your system, and those people reactivate their tuberculosis. So let's go back to GrandSim. What we're going to do now is add mechanism to GrandSim around the TNF story. So we build an entire model, 9 equations that describe the TNF dynamics from receptor, the molecules, receptor creation, binding, 2 different types of receptors for here, TNFR1, TNF receptor 2, internalization, degradation, TNF synthesis, all of these. And we're going to run this inside every single macrophage in our model. So now we're drilling down to the next scale. I've shown you a cellular molecular model. I've shown you a tissue model with the readout, and now I'm adding in a molecular detail intracellular model. And as I mentioned, it's 9 ODEs that have to run inside every one of those, plus tracking the diffusion of TNF at the cellular scale that has a feed forward and feedback between the models. And there's some probabilities that are very complex about how NF-kappa B activation happens in a macrophage upon induction with TNF. But if we do that, what we see is that TNF, the rate that TNFR1 internalizes is actually a bifurcation parameter, because on the left side here, you get clearance. When it's really, really small, you get clearance of the bacteria, but you leave behind a lot of inflammation signature. Maybe if it's a little bit larger, but still small, you get clearance, not even a granuloma left. Here, you get a granuloma, but it's able to really control very low bacteria. But if TNF happens too if that internalization happens too slowly here, then what you see happening is that granuloma can't control its bacteria and it's about to burst open. So now we've identified an actual target in the system, a mechanism that is a handle. Now you can use this as a drug target. If you can create a drug that can enhance TNF receptor binding and internalization, then you have found a way that you can improve people with TB outcomes at the granuloma scale. So this is how we can provide information to experimentalists with our model predictions. Okay, but we don't want this really complicated ODE model running inside every one of these macrophages, and there's thousands of them in our model every time we simulate this. So now that we got really smart by creating this model, we want to be able to shut it off and only use it when we really need it. When we're asking specific questions about TNF related things, we'll turn it on. But when we want it off computationally, we want to be able to be really smart about how we do that. So we call this method tunable resolution. So we have a paper on that, but I'm going to tell you a little bit about how it works. So tunable resolution allows us to fine grain and coarse grain a multi-scale model at will. You can even do it automated in your system. So it allows you to adapt to the degree of detail depending on what questions that you're asking. And it can assist you in model analysis, computational speed, and it can even help you debug your system. But I want to emphasize that it's not the same thing as model reduction. So in mathematics, there's this idea that you can create a complex model and you can dwindle it down to create an isomorphic model that is very similar in dynamics to the big one. But when you do that, it's mathematically the same, but biologically it has changed a lot. What tunable resolution does, it helps you preserve the biology that you're interested in while also simplifying the model. Okay. So I don't have time to tell you how that works, but I just wanted to tell you that we have a way to do that. We have a paper on it, and we're happy to discuss that with anybody that has questions. Also, we have a lot of numerical challenges when you link these hybrid models, and let me walk through them. Model linking, I mentioned to you earlier that that's a complex aspect. How fast your model runs, your numerical methods that you're using. So for example, in the last version of the model I showed you with the TNF submodel, we have the agent-based model scale, we have ODEs running in the system, and we also have PDEs that are describing the partial differential equations. These are different time scales that these things are happening. We have to exchange information at these different times. That creates lots of issues with these hybrid models. So I'm just going to show you one example here. It's with the numerical step. For example, the agent time step is larger than the diffusion time step that's shown below, where you would want to update the model and sync it at the different scales. This model needs to be synced larger time scale. This model needs to be synced at much faster time scales. And then you have to figure out ways to link up these syncing times. So that's part of the issue with creating these model linkings. Additionally, diffusion is always the rate limiting step in our models. And so we have used explicit methods, implicit methods, combined methods. Here's the three methods that we've studied the most: alternating direction implicit, like a Crank-Nicholson, fast Fourier, and alternating direction. And what we've done is run GrandSim under lots of different time steps and lots of different algorithms. And in the end, what works the best for us is this Crank-Nicholson method with a time step of 1 minute. And that seems to give us the most accurate outputs for all the different scales. So it becomes very important for your model to figure out what really captures the best and what works the best and what's the most computationally efficient. Again, these are really tough questions. We've been working on these things for years. So these are not trivial things, even though I'm summarizing them all in a single slide for you. Okay, so in just the last 5 minutes, I want to tell you a really neat method that we've developed on how to treat tuberculosis. So here's an example of a granuloma taken from a non-human primate. And what we're done is we treated it with one drug here and a different drug here, and we've looked at it under a microscope. And what you can see is that some of the drugs penetrate certain areas, this caseous dead tissue, and some don't. So here we're getting drug in, here we're not. Here most of the drug stays in this region, here most of the drug diffuses in here. Now, TB is very hard to treat because there's 4 drugs that you use, and you have to give them for 9 months. That's very patient unfriendly. The granulomas, as you can see, are heterogeneous, and that provides a barrier. And then, of course, I've told you that the bug grows very slowly. All these things together make it extremely hard to treat TB. So in our GrandSim model, we've added in a pharmacokinetic and pharmacodynamic model that runs inside. And of course, the drug enters through these blood compartments and then diffuses on the grid and is taken up by the cells. And there's a killing rate constant based on what we know about the PK and the PD of each of the 4 drugs that I'm going to show you in this example. So when we dose, here's just a weekly dose, a daily dose over 1 week. You can see here's the drug dose, and here's the bacteria populations in different areas on the grid responding to it. But what you notice is that the drug is below the C50, the killing concentration, most of the time, actually. So no wonder it takes 9 months to get rid of all the bacteria, because you're below your killing constant where it needs to be for the different bacteria types. So let's look at this. So here's the granuloma that I'm treating for 2 different drugs. I'm going to show you a drug dosing here. You can see that red is very high concentration, blue is no concentration. So the gradient is red to blue. And you can see that on the right here, INH, the drug, the half-life is so fast, it's already done. RIF is, on the other hand, still around, but it's diffusing, and most of the drug is outside of the granuloma. Hardly any is penetrating. There is the second dose. Again, you see a little bit gets in, but most of it is outside trying to penetrate into the granuloma, which is part of the problem of drug treatment for TB. So if you think about the regimen design space for TB, if you're trying to create a regimen that's going to be really optimal, there's so many parameters. There's 10 drugs. There's different segments you can give the drug, maybe one different type a month. There's different doses, different frequency, how many times a week you're going to give it. If you actually put that whole space together, it's about 10 to the 17th options. Well, that's even too many options for a mathematician to try to parse. So what we have to do is create an optimization to optimize the regimens. So we do that by creating objective functions that look at both dose and frequency, and we put it through our model, but in a special way. We looked at it using genetic algorithms and something known as surrogate assisted optimization. We thought GA was going to work for us, but I'll tell you why it was not the best fit. And so we turned to radial basis functions for surrogate assisted optimization, which means that you guess at the best fit for what might work there, and then you kind of optimize to the best fit. And so our objective function here is time to clearance plus dose times frequency. And when you use these radial basis functions, what you're creating is a surface, and you're asking to find the optimal on your surface. And so when we did it for genetic algorithms, boom, we got it every time right at the answer. And even if we relax the genetic algorithm, we get there pretty quickly. For the radial basis functions using this surrogate assisted optimization, we get pretty close, and we got there pretty quickly. I'm showing you here the average number of function evaluations though. It took 4,000 runs of GrandSim to hit that target spot with the GA. And even if we relax it, it took 350. With the surrogate assisted, it took 20. And as I told you, computational cost is a huge issue for us. So we chose to figure out a way to optimize surrogate assisted optimization to figure out what the best optimization approach was to get the best answer. Okay, so in summary, what I've told you is models are very complex, and it's very important though to build multi-scale models for each compartment and then figure out ways to link them together. I've showed you 3 different types of tools that we use to analyze them: uncertainty and sensitivity analysis, talking about the sampling procedures there, efficient numerical implementation and linking, and then tunable resolution as a way to sort of switch between coarse and fine grain. We also build our models and validate them continuously with data at individual scales, and then once we've linked the models together. And we usually start at the middle, what's most known model, and then build out from there. And then you can apply this really in any sort of setting. So this work is heavily collaboration with Jennifer Lindemann. I think all the work I talked to you about today, almost all of it has been in collaboration with Jennifer. And all of our wet lab collaborators are shown here. Veronique D'Artois and Joanne Flynn, Sriram. Here at U of M, we're working heavily on drug studies, and Brie Aldridge at Tufts. Here's a snapshot of our group having a joint group meeting with our wet lab collaborators. Jennifer's camera was off here. And then, of course, we've had generous funding from the NIH and from Bill and Melinda Gates Foundation and the University of Michigan for internal funding with Sriram. We have 3 grants together. So thank you. I'm happy to field any questions that you all have. I know there's a lot, but I'd also, since I'm at Michigan, I'd love to have you all reach out, look at our papers, ask questions, and happy to help any way we can with your modeling. Thank you.
Thanks very much, Denise. That's really impressive. That the scope is really staggering. I have lots of questions, but I'll be fair and be generous and see if there are other questions from the audience first.
Sure.
So Mariana, do we want them to put it into chat, or does Robert?
No, they can actually unmute
Okay,
themselves.
yeah, just
and
unmute
ask if,
and
yeah.
ask. Go ahead.
So it seems Robert has a question.
Hi, Robert. How are you?
Can you go back to the diffusion problem?
Yes.
To understand a little better.
We hate the diffusion problem, I'll be honest. All right, there it is.
And can you explain a little bit? Is it in 3D?
So that's a really good question. First, we only did this in 2D. All of these studies to date have been in 2D for the diffusion issue. Even there, it's been extremely complicated to sort of find the optimal method to use. And so the question is, how do you define what might be optimal for us? Well, there's a couple things. There's how fast it runs. There's also how accurate it is. And we do that based on our uncertainty analysis. So what we do is we run the model with this particular method and we say, what does it predict is the most important parameters in the model? And we sort of compared all of those. And sometimes it missed some of them and using certain methods, and sometimes it was able to identify them with other methods. So that's what we used as our comparison. Now, I'm not saying it was the best thing to use, but what we care about mostly are the outputs of the sensitivity analysis. So that's what we decided to compare. So that means we ran the uncertainty and sensitivity analysis with GrandSim using every one of these different methods for every one of these different DTs. And then we computed the outputs. And I think we had multiple outputs that we compared. And we said, which method always predicted the, all the right outputs. Does that answer your question?
Maybe just one more.
Yes,
please.
The FFT assumes periodic boundary conditions. Is that your preferred boundary condition, or is that just?
So that's a really good question. We don't have periodic boundary conditions in GrandSim. We have the boundary of GrandSim is a little bit complicated because we kind of choose granulomas that don't get too large. We actually try to stay very much away from the boundary. And as they get too large, we pretty much ignore those granulomas that we get that are encroaching. We have right now reflective boundary conditions as if, you're going into maybe the next compartment that would be there. So we use reflective boundary conditions in GrandSim, not periodic. But I don't recall what boundary conditions we used for the FFT. I'm guessing if you think it's periodic, then that's what we use there. But I can follow up on that and actually find out and let you know. Do you have any concerns about that? Because I'd love to hear them.
Well, I'm just if the physical problem requires some different boundary condition, then you might not choose the FFT. There might be some alternative.
And I think in the beginning we used FFT because it was the easiest for us to do. But as we continued, we found out it was not really up to snuff, which is why we moved to the alternating direction about 5 years ago. And then that guy is pretty clunky. So then we decided to go all the way implicit. But then we thought maybe we just better compare to see what we're learning here. And you're right, FFT always was the worst, which is why even here we ran it with a delta. We had to run it sometimes with a huge delta. So I think that's a really good point, and I'm going to dig back deeper on that and follow up with I'll follow up with an email, Robert, just to touch
Great.
back on
Okay.
that. Thank you.
Thank you, Denise.
Mm-hmm.
Thanks, Robert. Other questions?
I had a question about the sampling, and that you mentioned that you're about to publish.
Yes.
So that work, and then it's really not curious, but it's that the deterministic method works better than the stochastic. But might it be because of I don't know, for a large n, it works like the average of the other methods?
I'm thinking
like
that's exactly
mean?
right. That's exactly right. Because if you look here, you can see that the black line, which is the Sobol, is really the average of what you're seeing with the random and the LHS there. So I think that's exactly right.
Okay.
But it's, we haven't proven that. Maybe there's actually something out in the literature about that with sampling methods, which is not our area of expertise, but we use them all the time. So we thought maybe we better follow up and make sure we're using the best one,
Okay.
So
so that's what I thought. Maybe the mean in the stochastic mean tends to these Sobol sequences, and that's why you're seeing this behavior. Okay.
Yeah, and that's a really good point. I in fact, I'm sure we can because because we're still writing the paper, maybe we can actually calculate it and see if that's actually what it's providing us there. That's a great point, Mariana. Thank you.
Okay. Yeah, thank you.
So maybe I should get my question in. So actually I have two questions on two different two aspects, and they're of course both technical. At the very end, it went by a little quickly, but what is the nature of the surrogate model that worked best finally?
Yeah, that's a really good question. So this was work as part of Joey's PhD thesis that so do I have the slide here about what the model is? Yeah. So it creates the method actually creates a surrogate model. And I am this was from about 5 years ago, Krishna. So I don't remember exactly how it does it, but I know that it ends up with this surface.
I see. So you're sort of learning an objective function surface from the simulations, and
exactly right.
And then you traverse the surface. Yeah.
Exactly right.
Okay.
That's what it does. And which is very different, right, than what a genetic algorithm does.
Sure, absolutely.
So this method, it was quicker. As I showed you, but in the end, you know, GA is always the best. I mean, because that's what it's doing. It's learning. It just was for our purposes with an agent-based model, just took too many simulations to run.
I see.
Yeah,
I see. And do you know whether these surfaces? I mean, they look. They look pretty well rugged in places, but I suppose this is only you're showing us a snapshot.
Right, it's a snapshot of what they look like, but they're actually different depending on what the objective is, of course, because we can change our objective function to take into account other things. So it sort of changes. Here is dose and frequency, how often you give it, and what the dose is. And I want to tell you something. Joey did something amazing because, as I mentioned to you, you treat with 4 drugs for TB. So we had to start using Pareto fronts because you have to have multiple objectives simultaneously. And so really we're looking at the global minima, or actually they weren't global, they were local minima of Pareto fronts to get for all 4 combined. So it's actually quite interesting.
And these are also very high dimensional, right? I mean, you can never really see the you just take slices.
Right. So I'm just showing it to you here just because we could in this very simple example, but most of the time we never see the surface. It's all happening computationally.
Yeah. Oh, that's fantastic. The other question I had is related to your tunable resolution.
Okay.
Now, again, there too, you have I'm sure there's quite a great deal of complexity in terms of just how what part you choose to tune and what you don't, and even probably even the degree of tuning, right? I mean, you say tunable, but okay. The degree of tuning is also...
Yes, it is very vague on purpose because I will tell you guys,
but it's complex, right?
It's complex, and there's no rule of thumb. It's not like first do this and then do this. We give a heuristic for how to think about this, and we've had it run. It's so amazing. Even we've had it run automated in our model. So we had a model of a lymph node where we had all this complexity about how these cells interact with each other, but we don't need that all the time on. So the model actually says, do we need to run that? Or no. So it keeps running the simple version, and then it gets to a point, oh, we need the detail. It turns it on, and then it runs the more complicated version of the model, and when that's done, it shuts it off again. So you can think about it, for example, with the drug example I gave you. You could say, are we treating with drugs? Then we can leave that model off and just have this happening. But then when drugs are on, you might need a whole nother mechanism on, and so you would turn that on and have the model run with that on, and then shut it off when you're done treating or something like that.
And that too itself is an optimization. there's an optimization loop that you could put there. So okay, I'm going to say I'm going to bring up the terms that everybody has to at this time, right? So this thing could be learned also, right? So so you could turn loose machine learning, and actually even beyond that, right? You could even actually go to more general AI methods to try to really bring some other intelligence, right? It's not like there isn't a lot of intelligence already focused on this problem, but this is fascinating. So is that also being considered in either in your lab or in the field? Or?
Here's the thing that we have felt so far. and we're the only people that do this. Although some other people, they coarse grain their models, but I think the coarse graining is not done in or model reduction. It's not done with a pulse to the biology. And I think that's so important. So let me just say that we find so far that tunable resolution is every single model specific, because what you need to, because you're trying to preserve the biology, it's actually on a model by model basis. So here we were very focused on, when do we need this TNF submodel on or off? We also have models where we have the lymph node and blood compartments completely modeled in detail. And we decide, when do we need those whole compartments on or off, depending on, do we really want to focus on recruiting cells from the lymph node into these blood spots or not? We can just have a rate that cells come in, or do we really want to have all the complexity of what's going on in that lymph node compartment and the blood to travel those cells to the place? So we have found that we haven't figured out sort of a general heuristic for it, but that it's a model by model question that we need. Although I'm not saying it can't be done. There's probably a higher level perspective here that could ride above all this. We haven't gotten that far yet. Nobody pays us to do that. They pay us from NIH to do the biology, not to come up with these, so we need an engineer to step in, Krishna, and do this.
Sure. But NIH is all about, they're getting much more about AI and digital.
No, they are.
All those things, right? So, okay, now I should step back and see if there are more questions from the audience.
Can I ask a question?
Please go ahead, Anil. Yeah.
So thank you for the great talk. I have two questions on the optimization part.
Okay.
One, did you have any difficulties when you were formulating the objective function? I know it's maybe not super common to do optimizations in all of the fields. And then two, did you consider gradient-based optimization compared to a surrogate model-based approach?
So that's a really good question. We did not do the gradient based optimization. I'm sure of that, but actually for drug treatment, that may be something that's really relevant. So thank you for that suggestion. In terms of the objective function, we don't have trouble creating it. The question is, what needs to really go in it? For example, if you're developing drugs for TB to treat people in sub-Saharan Africa, cost is a huge area. And so for example, cost might be a really important piece that's left off of here. Also, what may be important is how long you can treat. The World Health Organization now has said we want to take it from 6 to 9 months down to 2 to 4 months. And we want to also decrease the number of drugs given, because again, that's a cost issue. There's also a toxicity issue. Do we add toxicity into our function? So there are many pieces. We're trying to really show what we have available as a tool to the NIH and to Bill and Melinda Gates, because these are the questions they're interested in. They have to tell us what belongs in our objective function, really at that point. But creating the objective function actually was not difficult for us. The bigger issue came when we were doing a multiple drugs. So we had to have the Pareto fronts where we were trying to optimize or minimize, in this case, 4 different drugs simultaneously. And we created this Pareto situation for that. So that's where the complication came in when we had to move to that. So yeah, there's a lot of optimization on top of optimization here, which is layering, and we're living in this space right now. So hopefully in a year or two, Jennifer or I could give an update and tell you how far we've come with this story.
Thank you for the insight. Seems like there's a lot of potential to bring in gradient-based optimization. I know there's some implementation costs, but now you can formulate all of the concerns as constraints and then have a single objective. But
yeah,
it sounds great. Thank you.
It's a great idea. I will look that up and maybe reach out to you, Anil, if I can't find some good ideas on that. Thank you.
Sure, I can send you an email to follow up.
Oh, fabulous.
To talk further. Yeah, thank you so much.
Other questions? Well, if not, we're well past our hour. Well, you all know how to find Denise, and I hope you will follow up, and certainly I intend to, because there is another question, Denise, so don't go off. I do have a much broader question that I do want to follow up with you on.
No worries.
Okay, great.
Thanks, everyone.
Yep, thanks everyone, and we'll see you at the next webinar.